Hi guys, so for this video we're going to be diving into the renal tubule a little bit more. So just to quickly recap the structure, this is the nephron, this whole thing. This is the renal corpuscle, which is the first part of the nephron with the glomerulus and the Bowman's capsule. The rest of this highlighted in blue in this video is the renal tubule. Now the renal tubule has the three parts. It's got the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. So I often shorten proximal convoluted tubule to PCT, distal convoluted tubule to DCT, and loop of Henle, LOH. Now, this dotted line that is going across here, this is showing you that part of the renal tubule is inside the cortex of the kidney, which, if you remember, it was the outer part at the top of the pyramids, between the top of the pyramids and the renal capsule, the membrane. And the other part is in the medulla. So the loop of Henle and the collecting duct, which would be here, is part of the medulla. And if you remember, the medulla has a striped appearance in those pyramids, and it's striped as a result of all of these tubes that are going downwards into it. The very important job that takes place in the renal tubule is tubular reabsorption. So if you remember from glomerular filtration, all substances that are small enough are squeezed out of the glomerulus into the cavity of the Bowman's capsule. So you will end up with glomerular filtrate here, which will have a lot of useful nutrients still in that glomerular filtrate. So tubular reabsorption makes sure that those useful substances are reabsorbed. They are removed from the tubule. Remember, anything that remains in the tubule will be lost as urine. So your entire renal tubule has capillaries that wrap around it, and those are called peritubular capillaries. So if I just show that here, you'll have these capillaries that come off the efferent arteriole, and they wrap around, so that all the useful things can be returned to the blood, anything that is taken out of the tubule. So these will all be peritubular capillaries, but I'll delete that for simplicity's sake. So when these substances are actively reabsorbed, active means it requires energy, and it's often going to be either against a concentration gradient, or you want to make sure that you are taking in everything that you need. So for example, you should never have glucose in your urine. If you have glucose in your urine, it means you are a diabetic. So it is important that even though glucose is squeezed out of the glomerulus into the Bowman's capsule, that glucose is all actively reabsorbed to make sure every single molecule of glucose is removed from the tubule and added back into the blood. So the tubule cells have two very important adaptations for this to take place. The first adaptation is that these cuboidal cells have many mitochondria. So these mitochondria all provide energy for active reabsorption. The other adaptation is that they all have microvilli. Microvilli are these finger-like projections. Now, they don't provide energy, of course. They instead increase the surface area for active reabsorption. So both of those are structure-function related traits. So once the useful substances have all been reabsorbed, it mainly takes place in the proximal convoluted tubule. Now we call this substance inside the renal tubule to be dilute urine. But I'm going to dive into the process that takes place in the loop of Henle, because the loop of Henle is very actively involved in the conservation of water. So I've just drawn part of a nephron. I've got a little bit of the descending limb of the loop of Henle. I've got the hairpin bend, and then I've got the ascending limb going up here, the distal convoluted tubule that follows, and then the collecting duct, which remember is collecting urine from many nephrons. There will be a nephron going off to the side there. So when tubular reabsorption is taking place, useful substances that your body might still need, like sodium ions, like glucose, like I said, chloride ions will follow. They will be leaving the tubule, and the proximal convoluted tubule, and then the descending limb of the loop of Henle. So at this point, you have your sodium ions leaving the urine. Remember, this would be called urine. You've got your chloride ions. And because you're lowering the water potential in the medulla, remember, this is the medulla. Anything below the line here is the medulla. What you're doing is you're making the water potential very low. So water will naturally leave by osmosis, because water will leave from where there is a lot of water that's still in the tubule over here. And then some of it will leave through osmosis, following the water potential gradient. But when we get to the ascending limb of the loop of Henle, there's still a huge amount of water that is left in this tubule over here. But what I've shown here with the dark line around the ascending limb of the loop of Henle, we say that it is impermeable to water. So what that means is that although there is a huge amount of water here, water is not able to leave the tubule and enter the medulla and therefore enter the capillaries because this wall is completely blocking any water molecules. It is impermeable, does not allow water to pass. But what the ascending limb of the loop of Henle does is it actively pumps out sodium ions. So this is active reabsorption, very, very important because a lot of energy is needed. Now, this would be why the cells have many microvilli and many mitochondria, so that this kind of thing can happen. But what happens by actively pumping out the sodium and the chloride ions, you make a very, very low water potential inside the medulla of the kidney. And what I mean by a low water potential, I mean it's a very concentrated solution. If you think about it, it would be like adding a cup of salt to 1 glass of water. Extremely concentrated, very, very salty solution. Now, that would be now what we call a solution with a low water potential. Whereas inside the tubule, all of the water that is in there has been left behind. So this is very, very dilute urine. Now, when this dilute urine gets towards the distal convoluted tubule, it travels here towards the distal convoluted tubule, and now the walls of the distal convoluted tubule and the collecting duct, for that matter, are permeable to water, meaning that water can now leave the tubule. Water will flow by osmosis because there is a very dilute solution in the tubule and a very concentrated solution in the medulla. So water molecules will naturally leave the tubule. Now, this ties in with the next video, which I'm going to make as a separate one, about how there is a hormone called antidiuretic hormone, which makes this tubule more permeable to water by opening up more water channels. And that is how the water is conserved in your body actively by your brain, by a control center. So please watch the next video that follows.